Hi, my name is Daniel Fernandez, and along with my fellow classmates, we will tell you all you need to know about the Kirby-Bauer test. So, first off, who are Kirby and Bauer? William Kirby and Alfred Bauer were research colleagues at the University of Washington, and throughout the late 1950s and 1960s, they proposed and published a method for testing the effectiveness of antibiotics, which are medicines that help fight off viruses and diseases in our bodies. The World Health Organization standardized it in 1961, and the procedure became known as the Kirby-Bauer disc diffusion test. Despite advances in technology such as automated tests, the Kirby-Bauer test is still used today to research different antibiotics. So what exactly is the Kirby-Bauer test? This procedure measures how susceptible bacteria are to different antibiotics. In other words, it determines if an antibiotic is effective at killing bacteria, which indicates if it works well at treating a particular virus. This is done through the use of discs that are imbued with or contain different antibiotics and are placed on a surface layered with bacteria. The way of telling whether the antibiotics are effective or not is through the zone of inhibition that is created around the discs. The way this works and how to examine the zones will be explained next.
Hi, my name is Natalia Daher, and for this project I will be answering how the Kirby-Bauer test works. For a little bit of context, the Kirby-Bauer test is an experiment done to determine how reactive or how sensitive a bacteria is to an antibacterial. We do this so that physicians and scientists are able to develop new antibiotics that can help fight different diseases and different bacteria. So in order to do this experiment, the first thing you're going to need is a little circular plate that contains your bacteria. So for example, E. coli. Once you have that, you're going to need little circular paper discs, and these are going to contain the antibiotic that we will put on the plate. But before we put them on the plate, we need to incubate them in order to filter them. We filter them for however long we need, sometimes overnight, sometimes a couple hours. Once you incubate them, you put them in an enclosed space for, let's say, overnight. You then divide your plate into different zones. Let's say you divide it into 7 sections, and you label each zone. And then you put your paper discs onto each zone. You put one disc in each zone. And then you wait for the results. You close your plate and you leave it there for, let's say, 1 week or a couple days or even overnight. And then you come back and you check your results and you measure your zone of inhibition. And the zone of inhibition, what this indicates is how sensitive a bacteria was to an antibacteria or an antibiotic. So there is a certain chart we have to follow to determine how antibacterial or not antibacterial the zone of inhibition is, or bacteria is. If the zone of inhibition is less than 6 millimeters, then it is not antibacterial. If it is between 6 or 16 millimeters, then it is slightly antibacterial. And if the zone of inhibition is greater than 16 millimeters, then it is highly antibacterial.
Hi, my name is Cindy Ricano, and today I'll be explaining why the Kirby-Bauer test is important for biology and everyday life. To recap what we previously learned in the video, the Kirby-Bauer test is used by scientists to measure the antimicrobial resistance against different bacteria. Whoa, what does that mean? Basically, how well the treatments work against bacteria. How well or how badly. Now, why is this important for everyday life? Well, in order to understand why this is important for everyday life, we first have to understand what is antibiotic resistance. So I'm going to give an example. Let's say every time you're sick, you take an antibiotic. Your body and the bacteria is going to keep mutating and adapting to that antibiotic so that it can fight against it. So the next time you take antibiotics, yes, the bacteria that has not mutated, it'll die. However, the ones that have adapted and mutated, they'll live and they'll keep multiplying, and then you'll stay sick. What can we do? This is where the Kirby-Bauer test comes into play. The Kirby-Bauer test measures the zone of inhibition for each plausible treatment against the bacteria. Whoa, that's a lot of words. Let's break that down. What is the zone of inhibition? The zone of inhibition, as explained previously in the video, it measures how well the treatment worked against the bacteria. So let's say you have a large zone of inhibition around the tested treatment, then that means the treatment worked because the bacteria isn't growing close to the treatment. But if you have a small zone of inhibition, the treatment didn't work too well. All of this is why the Kirby-Bauer test is important. Because of the aforementioned antibiotic resistance, we have to resort to natural alternatives as a substitute. The Kirby-Bauer test can compare the zone of inhibition for natural alternatives and drug antibiotics. whichever natural alternative matches the zone of inhibition best, like a certain antibiotic treatment, then we can use that to fight against the bacteria when we get sick. Thank you. Bye.